Good morning, everyone. The second experiment for this term is DNA isolation from onion. The DNA is the carbon copy of all organisms. It is a nucleic acid which is present inside the nucleus in eukaryotes. And it is a long polymer of deoxyribonucleic acid. And it consists of two polynucleotide strands which are helically wound around. And it gives rise to a double helical staircase structure which was proposed by Watson and Crick. In prokaryotes means the DNA mostly it is naked DNA. That means there won't be any histone wrapping around the DNA. And it is found within a single chromosome, which is single circular naked DNA. And the first attempt for extracting the DNA was done by Frederick Masson in 1869. So when we move detail into the structure of DNA, it consists of two polynucleotide strands. And the polynucleotide means many nucleotides. So the components of nucleotides are nitrogen base, pentose sugar, and phosphate group. So when we start with nitrogen base, they are coming under two major categories, that is purine and pyrimidine. Purine means it is adenine and guanine, which is double cyclic, which consists of carbon and nitrogen. The main cycle, two cycle will be there, carbon and hydrogen. Nitrogen will be there in that main skeletal structure. Pyrimidine means it is only single structure only, single cycle only. And it is of three different types, that is cytosine, thymine. And if it is uracil means that will be in RNA. So instead of thymine, uracil will be the base in RNA. Then the second component is pentose sugar. So pentose sugar, it is ribose in RNA and deoxyribose in DNA. That means in the second position, one oxygen will be less. So that forms the deoxyribose. Then the third component is phosphate group. And here the nitrogen base is linked to phosphate or to the pentose sugar, not phosphate, pentose sugar through N-glycosidic linkage. And that forms nucleoside. So sugar plus base forms the nucleoside. And the phosphate group is linked to the nucleoside through phosphodiester, through phosphate phosphoester linkage, and the neighboring nucleotides are interconnected by 5,3 phosphodiester bond. So in that way, many nucleotides, with the help of phosphodiester bonds, it forms a single polynucleotide strand. So it is only one nucleotide strand, polynucleotide strand in RNA means in DNA there are two complementary polynucleotide strands which is interconnected by the base pairs. So when we are saying the base pairs, the bases in one polynucleotide strand will be connected with the bases of the other polynucleotide strand, and there also the specificity is maintained. That is, they are anti-parallel. Anti-parallel as well as they are complementary to each other. So in one strand, the direction of phosphodiester bond is 5,3 means the other strand it is 3,5 direction. That is the word meaning of anti-parallel. And what do you mean by complementary base pairing means one side if it is the base is adenine, purine means the other side it will be pyrimidine. And that itself there is specificity. Adenine always binds with thymine with the help of 2 hydrogen bonds, and cytosine binds with guanine with 3 hydrogen bonds. So that is the the base sequence of one polynucleotide strand, if you know, means automatically you can decipher the base sequences of the other nucleotide strands. So these 2 polynucleotide strands will helically wound around, and that forms a double helical structure, which was proposed by Watson and Crick. So this is just a brief idea about the DNA, the structure, the base pairing and anti-parallel nature of the DNA. Now we'll move on to the next topic, that is the isolation of DNA from plant material. Here we can use any plant material. It can be banana or tomato or strawberry or vegetables like spinach or green peas, even onion. And here we are in the lab, we are using onion because it is easily available. So we can do it in the lab with the help of onion. And the aim is to isolate the DNA from onion. And here the principle is DNA. DNA is one of the nucleic acid which is found in all organisms, and it acts as the genetic material, and it is a carbon copy. And DNA, since it is negative in charge, negatively charged, it can be extracted from the plant material by 3 main processes. Because the plant material as such, it is made up of cell wall and cell membrane. So first we have to break the cell wall and cell membrane. So for that we can do the lysis step. So what do you mean by lysis? Breaking of the cell membrane and cell wall. So the lysis can be done by 2 methods. The first method is mechanical. So mechanical lysis is done by, in the lab we are using it with the mortar and pestle. So mortar and pestle, you can cut the onion into small pieces or small bits, and then you can crush it with the help of mortar and pestle. It can be even done with the help of blender. So but when you are blending it, you have to make sure that it should not be finely powdered. It should be only crushed. So the mechanical breaking can be done by the crushing or by blending the onion pieces in mortar or in blender. So the first step will be over. The second step is chemical breaking, chemical lysis, we can say. How can we lyse the membrane? By applying or make the permeability more permeability or more lysis of that cell walls and all, because it is plant material only, cellulose cell wall. So it will not be mechanically, it will not be crushed properly. So we are applying the chemical. The chemical here we are using is liquid detergent. Liquid detergent means the liquid type of dishwash, Vim solution, liquid solution, or even detergent wash liquid, which contain main ingredient, that is sodium dodecyl solution. Sodium dodecyl solution. So that with the help of this chemical, we can break finely or the complete lysis of the plant material, the cell wall, the cell membrane and all will take place, and then inside the components, even the nuclear membrane also will rupture. So the DNA will be can be taken out or will come out from the cell components. Then the next one, the requirements when we are using it, the next one's already the crushed onion with the help of mortar and pestle. Then the next one, it is liquid detergent, I told you, that is for chemical lysing. Then we can use the sodium chloride or salt, common salt we can say. The common salt will help in neutralizing the negative charge of the DNA. Okay, negative charge of the DNA will be neutralized with the help of sodium ions, which is present in sodium chloride solution. Then the next one it is, we have to use it is, that is chilled ethanol. So how can we make chilled ethanol? Means one overnight we have to keep the ethanol in the fridge, and it should be super cooled, and that type of chilled ethanol is used for precipitating the DNA which is already separated from the plant material. So when you are adding the chilled ethanol, the DNA threads will precipitate, will separate from the suspension, and then it will be floating on the top. So once it is separated, then with the help of copper needle or any pencil or any needle, you can spool it out. So you can extract the DNA threads or DNA strands from the onion suspension, which is mixed with alcohol and the liquid detergent plus sodium chloride. So this is the principle behind the extraction or isolation of DNA. Now the procedure I am explaining it, then we will do the demonstration. So first you have to take the available plant material. So in the lab we are using onion, or you can use spinach or pineapple or a banana, whichever the material is available, that you can use it, and you can ground it into fine, crush it, either ground it or crush it with the help of blender or with the help of mortar and pestle. So in the lab, it is mainly we are using the mortar and pestle. So with the help of mortar and pestle, the chopped onions can be made into or grounded into or crushed into pieces, small pieces. Then you have to fill take a beaker and into that 25 ml of water, and slowly add 2 spoons of liquid detergent. And to that you can add 1 spoon of half a spoon of sodium chloride, and gently you have to stir it. So if you are stirring it very fast, then what will happen? The froth will come out, the bubbles will come out, so that will disrupt or that will interfere the separation or the precipitation of the DNA. So slowly you have to stir it and make it into a solution. And to that you have to add the crushed or ground onion paste or the ground mixture into that, and again stir it thoroughly without allowing the formation of bubbles. Okay, that is very important. The bubbles will interfere with the result. Then keep that thing for about 5 minutes to settle, and also to separate, that separation of DNA or coming out of the DNA from the material. You have to wait or allow it to rest for 5 minutes. And after 5 minutes, what you have to do? You have to strain it, so that you can use the tea strainer, and which you can keep it on a beaker or in a vessel, and then strain it. The strained solution also you keep it for some time, because sometimes while straining there will be possibility of formation of bubbles again. So you can keep it for some more time. So you will get the onion extract. Then take a bowl, or you can take or a petri dish or even a test tube. So individually when you are doing it, test tube will give the best results. Or for demonstration purpose means you can use a petri dish or a bowl, glass bowl, then it will be easy. So take a glass bowl or a petri dish or a test tube, and to that half fill with chilled ethanol, the freshly chilled ethanol that is directly taken from the fridge. So chilled ethanol is for 45. It is very, very essential. So that fill the petri dish or bowl, half fill. Okay, half fill the petri dish or bowl or the test tube with chilled ethanol. Then you have to take the filler. With the help of filler, take the onion extract. And if it is test tube, means slightly slanted manner, 45 degree, you have to keep it. And with the help of filler, you have to drop the onion extract slowly into the test tube or petri dish or the bowl without mixing. Okay, without mixing, that means slowly you can add. So the junction between these two solutions will not be disrupted much. Then after pouring, that means half the solution, again the equal amount of onion extract you have to add, and without shaking you have to keep aside for about another 10 minutes. After 10 minutes you can see in this bowl, if it is petri dish, means within the petri dish you can see, on the top of the petri dish you can see white strands, cloudy appearance you can see. It will be somewhat in the form of a white strands. And if it is test tube, means that on the top you can see small strands, stringy appearance you can see, smoky stingy appearance you can see. That one will be the DNA strands. And if you have a needle or a loop, copper loop and all, means you can insert the needle or the loop and you can swirl. You can just rotate that one and the strands will get entangled and you will get the DNA strands. So this is the main procedure to extract the DNA from the onion. So the inference will be how it will be appear. So the DNA can be appear as white precipitate of very fine threads with the help of, on the spool. And the DNA can be isolated from the plant nucleus by this technique. So this is the summary of the theoretical part of this extraction. Now I will show you the doing part. How can we do by the demonstration of this experiment. Now we will do the demonstration. So the materials which are required: onion, then liquid detergent, that can be from any Vim or any dishwash or whatever it is. Then sodium chloride or salt, common salt, mortar and pestle, and knife for chopping the onion. Then chilled ethanol, strainer, then strainer for straining we need another beaker. The most important, it is chilled ethanol. It should be very chilled. Okay, so these are the main materials we require for that. First, take the onion piece, chop it into small pieces. So very finely you have to chop it and put it into the mortar. And with the pestle you have to crush the onion into pieces, small pieces. So make sure that it should not be pasted. Instead of that, it should be crushed properly. So even in, for easiest way is this onion pieces, we can keep it in a bowl of or jar of mixie and you can just crush it for 2 or 3 minutes. But in the lab, we cannot provide mixie or blender for everyone. So we used to provide the mortar and pestle. So you have to crush it and make sure that the whole plant tissues are smashed properly. So that's why the crushing procedure is a little bit lengthy. You have to crush it properly. Then after crushing, you have to take the liquid detergent, that is 1 or 2 spoonful of liquid detergent. To that you have to add half a spoon of common salt and stir it well. To that you can add 25 ml of water. So while adding water and stirring, you have to make sure that there should not be any bubbles, because quite naturally the bubbles will be there and that bubbles should settle down and you have to break all the bubbles, otherwise it will interfere the filtering of the DNA. And the crushed onion, after making the solution, detergent plus sodium chloride and plus water, to that solution the finely crushed. So that's why here it is not crushed properly. So that's why again I am crushing it and making it that the whole, at least the majority of the plant material, that is onion, is crushed and it is ground into a smoothie substance. So that crushed material is to be added to the solution. Again stir it well. And you have to keep aside for 5 minutes approximately. And make sure that that frothy appearance and all are disappeared. And if the solution is too thick, means you can add little more water to make it thin. And then only you will get a clear solution of the mixture. Otherwise, if it is too thick, means straining also will be difficult. And at the same time, the thickness of the solution also will be very high. So you can make sure that it is moderate thickness, and you can strain it with the help of a tea strainer. Then again you keep aside for few more minutes to make sure that the frothy appearance and the precipitation of the DNA and all will be taking place proper time. So after 10 minutes, you will get a clear solution of the onion extract. And after that, we can take a petri dish or a bowl or even a test tube. So students means usually they can use the test tube. And while demonstrating, teachers will use the petri dish. So first half fill the petri dish. Make sure that the onion is free from the bubbles. Then take the the ethanol, chilled ethanol, half fill with ethanol. First the bowl, fill it with ethanol, half fill only. Then to that you add with the help of dropper. So when you are taking the onion extract in the dropper, through the sides of this petri dish or the bowl or even the the test tube, through the sides slowly and gently only you have to pour it. When you are pouring it all of a sudden with much force means it will disrupt. So the solution that the junction between the solution should not be disrupted. So slowly through the sides, take enough patience, and then through the sides you can add the onion extract to the alcohol solution. Then allow it to rest for another 5 minutes without shaking. So when you are keeping it aside, make sure that it is not mixed by shaking it. Without shaking, slowly keep aside. So here I am showing how to add onion solution into a half-filled ethanol containing test tube. So first take the test tube, then half fill it with ethanol. After that, with the help of dropper again, take the onion solution, onion extract, and gently keep the test tube 45-degree angle. Then pour slowly with the help of filler or from the beaker, slowly add it. Don't mix the solution, don't shake the solution much. Carefully you have to keep on adding it. And then allow this mixture to rest for another 5 minutes. We can keep the test tube in a test tube stand for about 5 minutes. Then after 5 minutes, when you observe, we can see a cloudy appearance on top of this solution. So here in the petri dish, it is not clear that much because it is that density of the solution is not clear. But I will show you how it is separated in the test tube by keeping a darker background on this. Now you can observe a white strandy appearance on the top of that solution. So when you keep the test tube in a darker background, you can very clearly visible the strands of DNA. So now you can observe it, the strands of DNA. And once the strands are separated clearly like this, so very clear strandy appearance will be there. That is the DNA strands. Now you can see the DNA strands. By the spooling technique, we can extract in this way the DNA strands can be separated.